Welcome to Monkeys on Typewriters on YouTube. We're still talking about the topic of cancer and evolution. You may recall last time Arijit gave us an overview of how chromosomal instability is central to the development and evolution of cancer. Now, in part two, he's going to follow up talking specifically about oncogenic viruses or viruses that cause cancer. So I will hand it to you, and let's follow up with this topic.
Sounds good. Can you hear me?
Yep.
Great. Yeah. So let's get going. Thanks for the intro, Ryan. Nice to see you're back again for part two of this. Hopefully you'll hold up as we get into more dark and disturbing territory. But so off to the races we go. So let's talk a little bit about what we know about oncogenicity as far as viruses go. So there's a number of viruses that have been sort of widely accepted to cause cancer. EBV is one of them. Hepatitis B virus is another. HTLV1 is another. And then you have things like HPV, hepatitis C virus, and Kaposi sarcoma virus, which is something that you see in AIDS patients, and Merkel cell polyoma virus, which is the most recent one. Now, the funny thing about this is that people have been talking about viruses causing cancer for a very long time, for nearly a century. But only in recent years, like from the 1960s onwards, did people actually start making that association. And the definitive associations of these viruses with cancer took decades. So it's really been literally in the last 30 years or so that it's well acknowledged that a number of these different viruses actually are the causal agent for cancer. And that controversy has sort of died down. But it's kind of funny because we talked about viruses being carcinogenic for a long time. The first oncogene was actually RSV was one of the first oncogenes identified. Rous sarcoma virus led to the identification of RAS protein, which is one of the first oncogenes. I misspoke about that, but yeah, but that was a chicken, that was a chicken thing. So it took a while for people to really zero in on these viruses being capable of causing cancer. And a large part of that is because they were focused on oncogenes. So, with the RAS oncogene, RAS and SARC and many of these other oncogenes that are sort of well known in signaling and have signaling roles in cellular tissue, people started thinking about, okay, well, let's go looking for viruses that have oncogenes in them. And they, in fact, did find these, right? So EBV has an oncogene in it. HTLV1 famously has the TAX oncogene. So there are all these different oncogenes that have been identified. And so it's a very, very oncogene-centric way of thinking about it. Now, what happened, kind of like a funny thing that happened along the way, is not every single one of these viruses has an associated oncogene. So, for example, hepatitis C virus causes cancer, and people have looked 6 ways to Sunday, but they haven't found an oncogene in this. So, a little bit of a wave of the magic wand and some special pleading going on in this diagram here, as you can see. And so they say on that one, they say chronic inflammation. So, I mean, it's all fine. Is it true that these viruses cause cancer? Yes. Is it true that they have genes within them that promote the causation of cancer? Yes. Both of those things are true. But we'll talk now in more concrete terms about sort of how you can look at this from the lens of, as we discussed last time, chromosomal engineering, and see how these things are subtly different, and then they have big implications going downstream. So Merkel cell polyoma virus is one of the latest viruses that has been identified as being oncogenic, literally, I think, within the last decade. And so the virus is actually a reasonably, sized virus. It's 5.4 kilobases. And it's got, quite a bit of protein sort of infrastructure in there. And what they found was this protein VP1 functions as an oncogene. So it interacts with proteins, and what it ends up doing downstream is it ends up transforming cells. So they were able to find that, okay, the VP1 oncogene alone serves as is sufficient for transformation. So what they found was, when they sequenced, when looking for Merkel cell polyoma in these actual polyomas, polyoma virus, they found that pretty much most of these had Merkel cell polyoma. The polyomas had the virus in them. And then this virus, viral T antigen, was what they found in each case. So at the end of the day, what they were able to identify was, okay, well, the knockdown of the viral T antigen ablates cell proliferation. So you could make the argument that viral T antigen oncogene expression is addictive, and so the virus is addictive to this viral T antigen. This is from their paper, right? And so this is kind of how people talk about it when they talk about Merkel cell polyoma virus being an oncogenically driven oncogenic virus. But last time we had discussed the evolutionary paradigm of cancer. So what we had discussed is cancer is not many different diseases. It's one disease, and it's a process of stochastic Darwinian evolution with a single somatic Darwinian evolution with a single initiating mutation or a couple of initiating mutations, particularly mutations that initiate chromosomal instability, which pretty much every single oncogene does, and mutations that prevent downstream checkpoints that can arrest cell cycle progression in the presence of chromosomal instability. And those mutations are usually found in tumor suppressor genes. So while it sounds similar, because we're still talking about oncogenes and tumor suppressors, it's fundamentally different because this is a stochastic, not a deterministic process. The stochastic accumulation of mutations, and within an individual tumor and within an individual patient, the life histories start becoming very divergent very quickly. So not only does every patient have their own unique natural history, but actually every lineage of every clone of cancer cells within the patient is its own lineage. So in this particular case, also genes aren't driving the cancer; they are being shaped by selection pressure. So unlike the oncogenic model where you say, okay, well, the oncogene has to be present in order for the cancer to be moving forward, right? It's thought of as the accelerator, and the tumor suppressors are thought of as the brakes. In the chromosomal instability model, it's much more like releasing the handbrake if you're parked on a hill. Once you release the handbrake, you start rolling downward. Pull that handbrake up all you want. It's not going to fix the problem. So when you think of chromosomal instability, cancer being caused by chromosomal instability, it's more that latter model. Now, let's look at Merkel cell polyoma virus again. And we talked about the T antigen being sort of Merkel cell polyoma being addicted to T antigen oncogene expression. There's a different way of looking at this because T antigen actually does something in these cells. What does T antigen do? It doesn't have some sort of, it's not that it has some sort of, if you will, undefined or inchoate drive towards proliferation. What's happening is that small T antigen protein expression leads to multipolar mitoses. And those multipolar mitoses you can see on the left. So in this particular case, you see these cells. The blue is the chromosomes, and these are cells during mitosis. So you can see they're at metaphase. All the chromosomes are lined up neatly. The chromosomal poles, the spindle poles are marked in red. And that's rhodamine, I think, marking the tubulin. And on the right-hand side, you can see what happens in an overexpression situation. So if you overexpress MCP small t antigen, you see multipolar mitosis. So what ends up happening is that you have in this case, three different poles, and those chromosomes are not going to segregate properly. So then you see on the right-hand side, you see aneuploid divisions, which are greater than 4n. And you can see that there's a small number over here. You get these essentially polyploid cells, roughly polyploid, but not exactly. And you can also see that there's a little bit of a change in the content between 2n and 4n in this FACS profile. So all the 2n cells are here. The 4n cells, which are getting ready to divide, are right here in a normal situation. With the overexpressor, what you see is the DNA content. You still have the 2n and 4n cells, but now you have all these cells that are kind of a little bit polyploid. They're quantified on the right-hand side. So that polyploidy is enough because that polyploidy, as we talked about, that aneuploidy leads to what's called micronuclei formation. And they quantified that in mice now, not in cells. They had overexpressing mice for the small t antigen. And what they found was that mice overexpressing the small t antigen in the presence of CRE end up, which allows it to be expressed, end up with a higher frequency of micronuclei. So what you can see over here is that you're ending up with aneuploidy. Now, once you have aneuploidy, then that sets in motion steps of carcinogenesis. That's true for all categories of carcinogens and for viruses as well. So we talked about Merkel cell carcinoma as a model for oncogene addiction with this multi-antigen. And we're also looking at Merkel cell carcinoma through a different lens, through the CIN model of carcinogenesis. So when you look at it through the chromosomal instability model of carcinogenesis, it's not that anybody's debating the fact that multi-antigen causes cancer, but it's a how that we've reframed, or we're viewing a little bit differently. What we're saying here is it causes cancer because it causes multipolar mitosis, and it causes chromosomal missegregation or chromosomal instability. And that missegregation of chromosomes, where the chromosome count in each cell is different over time, is basically a driving force for heterogeneity, genetic heterogeneity, which is leading to evolution. So it's actually somatic evolution in action. Now you could say, well, isn't that all semantic? Like, what are the practical consequences of this? And one major practical consequence is if you're looking at a model where you have an oncogene, if you're using the oncogene mental model, the oncogene has to be present the whole way through. If you're using the chromosomal instability mental model, first of all, you don't even need oncogenes, but the oncogene doesn't actually have to be present. Over time, you can actually end up losing that oncogene. So this is an updated model of viral carcinogenesis that someone put out there. And here they kind of, they're including genomic instability as, one of the possible drivers. It's still overly complicated. Like, this is not exactly what we're saying, because we're talking about this as a stochastic process of evolution. But whenever you actually try to draw the stochastic process as a set of neat arrows, you're going to get something like this. It's going to look super complicated because obviously there's many different ways in which these things can happen, and they can happen out of sync with each other, which is kind of what you see in this highly complicated diagram. But the take home is still the same, which is once you have a persistent infection, and once you have the induction of genomic instability, then you get essentially you're off to the races. You get cell death, you get proliferation, and you get the progression of the cancer. So in a certain way, the arrows over here don't exactly line up with how you would view this from an evolutionary perspective, but all the elements are very much here. As you can imagine, it's very difficult to assign causality in a situation where you have a stochastic process of somatic Darwinian evolution. What's coming first, what's coming second is a little bit more up in the air, if you will. But the important point is that once you have somatic Darwinian evolution going on, it's off to the races for that tumor, because that collection of cells is now playing for itself, optimizing its genome and working out sort of strategies to keep proliferating and to keep blowing past the body's checks and balances, both cellular checks and balances within the cells themselves, and also external checks and balances at the tissue level or at the level of the immune system. So in fact, it turns out that most, if all, viruses also cause DNA damage and suppress the DNA damage response. And that's a consequence of chromosomal instability. Once you have chromosomal instability going, you end up with a lot of double-strand breaks. Those double-strand breaks are both causal in chromosomal instability, and then they promote further chromosomal instability. So on the left-hand side, you see this is a diagram that shows the activation of the DNA damage response by virus replication and by specific viral proteins. And what you can see is that a lot of the proteins that we've talked about, such as the SV40, the T antigen or HTLV1 tax, are involved actually in these DNA damage responses. And at the same time, these are also involved in suppression of DNA damage response signaling. So it sounds paradoxical that you would want both induction of DNA damage and then suppression of DNA damage response signaling. But not really, because the induction of DNA damage is what gets you going on chromosomal instability. Because once you have DNA damage, you start getting chromosomes broken into what's called micronuclei, and those broken micronuclei drive chromosomal desegregation. We discussed this at length in the previous lecture. So what we're doing is we're just going over that at high speed now. But if you want to learn more about it, we went into it in depth in the previous talk. So the thing about that, though, is that once you have that, the initiation of chromosomal instability, checkpoint failure allows those cells to proceed without blocks on their replication, and that's what essentially puts in motion the process of somatic evolution. So that combination of sort of double-strand breaks and cellular DNA damage response is highly characteristic of oncogenic viruses or carcinogenic viruses. Here is one figure for HTLV1 tax protein. We're going to go into this in a little bit of detail. For those of you who are not into this kind of level of detail with biochemistry, feel free to skip a few slides ahead. We'll point out sort of where to get to. I think we will be at slide, let's see. We're going to be at slide 53. So you can join us again when we get to slide 53. Look at the bottom right here, the bottom right for the slide number. But so we're going to go into some slightly nauseating detail now about sort of what we're seeing. So there's several different ways you can look at DNA damage. You can do the Comet assay, which is in figure A over here. You can stain with immunofluorescence microscopy for gamma H2AX, which marks the point of DNA double-strand breaks. It's a protein that actually sits on those double-strand breaks. And you can also look at gamma H2AX in panel B and C. Sorry, in panel C, you can look at gamma H2AX with Western blotting, where you grind up the cells, and now you're looking at that band. And the presence, the darker the band, the more gamma H2AX you have in that cell. So what you can see over here, first of all, is that HDLV tax induces a double-strand break. So the controls are on the top left. And then what you see over here is that HDLV1 transformed cells show double-strand breaks. And you can see that over here in these, in the panels, let me just show it here. It's in these panels. You can see the double-strand breaks. The reason you see them is because these cells are moving under an electric current. And so normally, if there are no double-strand breaks, they'll just move in as one mass. But when you get double-strand breaks, that DNA starts smearing out. When you visualize it, you can see it. So those comet tails in this assay are actually directly broken DNA. That's what you're seeing over here. So you can see that HDLV infection gives you DNA double-strand breaks by the Comet assay. You could also see tax-only cells transfected with the tax protein, showing you those double-strand breaks in these three panels over here. So what you can see is that both HDLV as a virus and the tax protein alone generates these double-strand breaks, as visible by the Comet assay. You can also see that over here, that cells transfected with the virus alone show you gamma H2AX double-strand breaks, as well as cells transfected with tax, which you can see over here. These little dots are the actual double-strand breaks. You don't see that in the control in the top panel over here. They've quantified it on the right side, over on this side. So panel C over here shows you, again, the DNA double-strand breaks. You can see them via Western blot. And the band over here is the gamma H2AX, which is the form that binds to DNA double-strand breaks. You can see that while the control cells don't show it at all, you see it in both the tax transfected cells as well as in cells with an infection, with an HDLV1 infection. So there's more to this, but just that kind of summarizes it for now. So you're looking at three different ways. In this case, with HDLV1 tax, you can see the DNA double-strand break response. So not only do you have the double-strand break response, but it actually subverts the double-strand break response. So when they looked at, in this case, what they did was they infected cells, and then they looked at tax expression. And what they were able to transfect and looked at tax expression. And what they found was that when they took tax transfected cells and now irradiated them, they found that that what ended up happening was that the DNA damage response in this case was altered. So what you can see over here is these are basically, again, immunofluorescence assays. Blue is the DNA, DAPI. Red is gamma H2AX. You can see these cells are getting really bright with a ton of DNA damage. And green is the cells that have tax transfection, so the oncogene transfected. So what they did over here was, what they show over here is that the cells that have the oncogene in them have more DNA damage. You can also see that over here in the Western blot, where the oncogene is present with irradiation, which you can see on this side, you get a more pronounced DNA damage. But the funny thing is you also get that without irradiation as well. So what you're seeing is you're seeing altered formation of DNA damage response markers. People have quantified HTLV tags in a different paper. They looked at micronuclei directly, and what they showed in this paper was when you have HTLV tags transfection, you get a bump up in the presence of micronuclei, which you can see over here and over here. So again, this is an example where people were looking at micronucleus formation, and they were able to see that in the presence of HTLV tags, you end up with higher levels of micronuclei, which is exactly what you would expect. So relevant to cancer biology, this is a paper from the year 2000. They point out that our findings here help to extend generally the idea that retroviral oncogenes, like mutagens, may work through heightened genomic mutability to affect transformation. So this is a paper from 25 years ago that's essentially making the point about the chromosomal instability model of carcinogenesis using HTLV1 tax protein as their example. Now, Kaposi sarcoma virus also causes DNA double-strand breaks and in this case, impairs the DNA damage response. So you can see that over here. Once again, you see gamma H2X Western blot staining with and without the Kaposi sarcoma virus present. And what you see is in the presence of the Kaposi sarcoma virus, you end up with an induction of DNA damage response, as can be seen by both by the Western blotting as well as by the Comet assay. And you can also see that with, then on the right-hand side, you can see that when it comes to non-homogeneous end joining, which is a form of DNA damage repair, that DNA damage repair is compromised in the case of, in the presence of Kaposi sarcoma virus. So again, the infection of Kaposi sarcoma associated herpes virus both induces the DNA double-strand breaks and also negatively impacts double-strand break repair. So as we've discussed before, once you have double-strand break repair, then you're you're at risk of forming micronuclei and chromosome missegregation, because those broken pieces of DNA are very difficult for the cells to align properly on their metaphase plate during mitosis. And that is exactly what you see with Kaposi sarcoma virus. Again, over here, what they show is, this is a different paper, and what they show is essentially induction of chromosomal instability with primary human endothelial cells. So when they infected cells with Kaposi sarcoma virus, what they showed was essentially the control is on the top panel over here. DAPI, the DNA, is on the left. Gamma alpha tubulin, which marks the mitotic spindle, in the middle. And what you can see is the controls look, this is what the controls look like with two poles. And what you can see with these multipolar mitoses is that basically once you have KSHP infection, essentially it kind of, all hell breaks loose. You can see that on the left. You can see that on the right as well, in terms of abnormal numbers of centrosomes, meaning abnormal numbers of spindle poles. So there's obviously issues with division. What they found over here is the effect is quite dramatic. It goes up from like less than 2% or in fact, less than 1%, all the way up to like 7% of mitotic divisions showing abnormalities. So obviously with those kinds of abnormalities in mitosis, you can imagine that there's going to be trouble downstream, which is what they found in this paper. So what they did was they looked at cells in anaphase when the plates of chromosomes begin to pull apart and towards their own two daughter cells. So during anaphase, normally a normal anaphase will have two plates of chromatids kind of pulling apart like this neatly, and then ending up in the daughter cells. And what you can see over here is that's not what happens. So again, A is the control. You can see what an anaphase plate should look like. And during anaphase, you should have these two sets of chromosomes pulling away neatly. What they found was that there were many cases with KSH3 like this, where you can find crud in between them. That crud is actually lagging chromosomes during anaphase. Now, those lagging chromosomes can often get trapped in the cleavage furrow and can end up leading to micronucleus induction. So that trap in the cleavage furrow is called mitotic bridges. And what they were able to show was both chromosomal laggings going up as well as mitotic bridges. They were able to find both in this case. And again, those are dramatic increases. Now, that begs the question, okay, if that's happening, then would you see micronucleus formation? And so they went ahead and quantified that. And indeed, they found that, again, this is on the top panel is what it should look like. These are individual nuclei with DAPI. And on the bottom panel, you see what happens with KSHV virus, and you can see there's multiple little micronuclei forming. Again, it's a dramatic increase. So In other words, Kaposi sarcoma virus, which is considered to be an oncogenic virus, has oncogenes present, is again causing chromosomal DNA double-strand breaks, chromosomal misalignment during metaphase, lagging chromosomes in anaphase, micronucleus formation. It both induces separately, we're not getting into it here, but it both induces and then suppresses the DNA damage response. So actually, we did discuss that a few slides back. We saw double-strand breaks being formed and also the non-homologous end joining being impaired. So all of the elements required to get, somatic evolution going, jump-started, is present in cells infected with KSH3. So again, that's what makes it an oncogenic, a carcinogenic virus. So this is getting, this will get old fast, but HPV does the same stuff. You can see again, double-strand breaks here, measured with gamma H2X on the right-hand side. You can see an alteration of check1 and check2 signaling, which are DNA damage proteins. What you can see over here is that HPV infections cause DNA double-strand breaks, and then they also induce checkpoint signaling. There's a specific protein in HPV called E6, which is an oncogene. And sure enough, that oncogene, as you would expect, causes segregation errors. You can see the control on the top left over here with no error. And then you can see all of these different kinds of errors, which is mitotic bridges, acentric chromatids lagging during anaphase. Each one of these panels shows a single pair of anaphase plates within a single cell. And you can see this is what it should look like over here. And this is what it looks like in transfected cells. In each of these cases, you can see that it's not happening cleanly. There's chromatids being left behind and there's kinetochores being left behind. You can see the number of bridges being formed. You can see the number of segregation errors. And what you can see is that this oncogene, again, drives up the number of segregation errors, as you would expect. You also see a dramatic rise in the number of anaphase bridges. So HPV E6 protein also induces micronuclei again. In many cases, there are multiple papers on these topics. I'm just giving you sort of an overview of them really quickly. What you can see over here, this is a fairly busy slide, but what you can see is the transfection with HPV6 protein leads to an induction of micronucleus formation. And you can see it over here on the top right. That the left is a control, and then here's your induction of micronuclei. What they show over here is that micronucleus formation is associated with centromere positive micronuclei as well. So centromere positive micronuclei are basically micronuclei that have taken like that chunk of the chromatid with it. So it turns out that oncogenic viruses all follow similar behaviors. So what you can see over here is this is a paper that again talks about this from a review perspective. And they show you sort of with these different proteins, EBV, EBNA1, which is an oncogene, HPV E6, which we talked about, and HTLV1 tax, which we also talked about. What you can see over here is that they talk about all of these proteins, oncogenes, leading to genomic instability. And again, this is a complex diagram because what they're trying to do with this diagram is they're trying to put like a, if you will, a deterministic view or a circuit diagram view on what is essentially a stochastic process, which is evolution. These things don't actually function in this way as neat little arrows where you go from one state to the other neatly, right? There may be different populations within the different cell lineages within the population that are doing different things at different times. And when you actually watch these things, as I have under video microscopy, when you watch cancer cells responding to treatment, it's a mess. One lineage will be doing one thing on one side, one lineage will be doing another. For a chunk of my graduate school and then my early career as a scientist, I used to do a lot of video microscopy of cancer cells under treatment. And we would look for, common themes, and there are no common themes, right? Because different cells are doing different things at different times. Because actually, that's how I got interested in evolution. You can actually see it when you watch a video microscopy movie of a set of cancer cells under treatment. We had actually discussed in the previous talk, people have actually sat and quantified those video microscopy movies, and they've shown that actually what you're seeing in those movies is that different lineages of cancer cells have differential survival, exactly as you would expect with evolution. We our cells showed that, and I showed some of that work in the previous talk. We showed that as well with a different kind of system where we looked at individual clones growing on soft agar, clones of cancer cells growing on soft agar, and we showed that they had very different growth kinetics under treatment, suggesting again that evolution was taking place. So again, this is a deterministic view, if you will. It's a circuit diagram view of a stochastic process. They don't have, these guys are traditional biochemists, so they're not representing what's happening through an evolutionary lens. But again, you see all the elements here. You see the DNA damage responses, you see the cell cycle arrest, and then you see at the center of it all, genomic instability. So as we've discussed, and again, this is just a small sampling of what's out there in the literature. There's a ton of papers, and actually in this presentation, there's a lot more examples. When we write this up in a more formal setting, we'll put all the references in, because each one of these points is made often with multiple papers. And I was showing you a figure here and a figure there. So what you got was a grand tour, if you will, of this table. And what the table shows you is the oncogenic viruses, and what it shows you is the formation of double-strand breaks, the engagement or suppression of the DNA damage response, and the formation of chromosomal instability, as mentioned often by micronuclei or by other markers such as anaphase bridges or lagging chromosomes. And what it comes down to is every single one of these viruses has been reported to do all of these three things. And as I said, in many cases with multiple papers. So this is not all that controversial in the literature. You can find there's a sheaf of papers, which as we start moving towards the written versions of these things, we'll be sharing the references on. But hey, go ahead. If you feel skeptical, go to PubMed and type in one of these virus names, and then type in chromosomal instability and see what you find. So there's a downstream consequence of chromosomal instability, which is that you get micronucleus formation. And one of the early cellular defenses for micronucleus formation is basically the cGAS-STING pathway. So that gets activated during viral infections as well. And we had talked about this in our previous talk. But the point is, we had talked about it as a general consequence of micronucleus formation and chromosomal instability. The point is that it is activated during viral infections. Multiple different papers have shown this quite compellingly. And viral proteins have been shown to antagonize cGAS-STING protein. In fact, SARS-CoV-2 has quite a bit of machinery in place to shut down cGAS-STING machinery, for example, as a virus. But many others do too. HIV, HTLV, HPV, they all have proteins that interfere with cGAS-STING signaling. So there's also indirect methods of viral carcinogenesis, such as chronic inflammation and lymphopenia. Both of these have been shown. In particular, chronic inflammation has been shown to lead to DNA double-strand breaks, and then from DNA double-strand breaks onward to chromosomal missegregation, micronucleus formation. So this is an important point, which is, for example, with hepatitis C virus, using a chromosomal instability framework lets you sidestep this entire question of whether or not there are oncogenes, because hepatitis C virus doesn't have oncogenes. And so as long as you have those DNA double-strand breaks being formed and DNA repair being suppressed, you can be off to the races in terms of induction of chromosomal instability and carcinogenesis. It turns out that viral carcinogenesis is also facilitated by various types of immune suppression. So there's many different kinds of immune suppression that these oncogenic viruses cause. And you can see over here, here they are. And the point is that many, but not all, oncogenic viruses modulate the immune system. And that actually facilitates that process of cancer induction, as you can imagine. I'm not going to go into the details, but again, this is very well documented in the literature. And the point is that once you have that sort of modulation of the immune system, it facilitates the development of tumors, because a lot of the early roadblocks to tumor formation really are placed in first at the cellular level and then at the level of the immune system. But it's important to keep in mind that immune suppression plays a variably important role in carcinogenesis. It's helpful, but it's neither a necessary nor a sufficient condition. So as you can see over here, there's a wide variety of different cancer causing viruses. And you can see they have a column with role of immunosuppression. And you can see it's all over the map. It's not necessary. Viral infections, whether or not they're oncogenic, suppress apoptosis. This is well known. So, here's a paper that shows a number of different viral proteins and how they impact different aspects of the apoptotic cascade. Why is suppression of apoptosis important? It's important because when you have cells that have abnormal numbers of chromosomes, or when you have cells with unrepaired DNA double-strand breaks, if any checkpoint signaling is present at all, there'll be sort of a push towards getting those cells out of there with apoptosis. And being able to suppress that allows viruses to take those, provides a fitness advantage for the viruses because it allows them to keep those cells, which are still churning out viral particles, productively engaged and sort of keeping them from being removed. So from the virus's perspective, it makes a lot of sense. But hey, it also has the side effect of promoting cancer, which is what ends up happening with viruses that suppress apoptosis and are oncogenic at the same time. Now, another interesting point is that oncogenic viruses often persist within the body. It really helps a lot if you're planning to create trouble too. It helps a lot if you can stick around. Now, what you'll notice is that these oncogenic viruses listed over here are pretty much almost always persistent. HPV is not always persistent, but if you have a persistent HPV infection, we'll talk about it another time, but your risk of carcinogenesis goes all the way up. So having a persistent HPV infection is really bad news when it comes to oncogenesis or when it comes to cancer risk. Now, note that for many of these oncogenes, there's an incredibly long, oncogenic viruses, there's an incredibly long lag time between actually getting the viral infection and showing up with cancer. And I'm just saying, if we look at COVID today, or we look at COVID-19 and we say, oh, we're pretty sure it doesn't cause cancer, you have to keep in mind that you haven't yet done the time, in order to allow you to draw that conclusion. So those people who say that we're pretty sure that it's not happening, we don't see any data for it, you can't even make that point until you've waited, 10 or 20 or 30 years. So we'll get more into that at another time. But the important point here, the other important point here is that persistence is really critical. Now, not only are oncogenic viruses persistent, but they're persistent in exactly the wrong tissues. And what are those tissues? The tissues that are dividing rapidly. So pretty much every single oncogenic virus, it so turns out, and this is a table I made, again, the references end here, and we'll keep adding more references as we get to the written version. But it turns out that every single one of these oncogenic viruses, in fact, infects dividing tissues, which is kind of really handy if you're trying to get a cancer going, right, from the virus's perspective. So EBV infects B cells and epithelial cells. HCV infect hepatocytes. HPV infects certain specific epithelial cells that are actively dividing, as does KSHV, which is Kaposi's sarcoma virus. Many of these viruses also infect immune cells, lymphocytes in particular. And Merkel cell carcinoma, weirdly enough, infects this one dividing cell in the skin called, not surprisingly, Merkel cell. But so it's kind of interesting that not only are they persistent, they're persistent in dividing tissues, and then they go on to cause cancer in the tissues that they infect persistently. So, all of this ties together pretty neatly. And so this is the last slide in this section. So you can devise, if you will, a checklist for viral carcinogenesis. And what it is does the virus cause persistent infections? Does it infect dividing tissues? We showed that every single one of the oncogenic viruses does in fact do these things. And then do the cells that are infected with the virus experience insults that we discussed previously in the previous talk that are known to be carcinogenic? And those insults are elevated levels of DNA damage, specifically unresolved DNA double-strand breaks, and micronucleus formation. Also, prior to that, missegregated chromosomes, abnormal mitoses, or abnormal mitotic spindle formation, anaphase bridges, or mitotic bridges at telophase, which we also saw, leading ultimately to micronucleus formation. As we had discussed in the previous talk, there's many different ways to get to micronuclei. Once you get to micronuclei, though, you have chromatin hanging out in the nucleus, in the cell that's not part of the nucleus, you have ongoing chromosomal instability. So micronuclei are both a contributor and a hallmark of chromosomal instability. And every single one of the viruses that we talked about today induces micronuclei. We also talked about secondary mechanisms that facilitate tumor initiation, progression, and these oncogenic viruses have those secondary mechanisms to varying extents. Not all of them have them, but obviously the more of these you have, the more helpful it is if you're trying to make that kind of trouble, if you personally are a virus and listening to this talk. Suppression of C-gas signaling, suppression of apoptosis, lymphopenia, and chronic inflammation are all useful tricks to have in the toolbox if the goal is to cause cancer. As I said, if you as a virus were interested in that topic. So again, the first part of this, causing persistent infections, infecting divided tissues, and causing these specific insults that lead to chromosomal instability present in every single one of the viruses that we discussed and that are known to be oncogenic. In fact, there are viruses that are not currently accepted as being carcinogenic that cause all of these insults. What are those viruses? We'll discuss that. We'll discuss one of those viruses next time. And if you've been paying attention to Ryan and my Twitter feeds, you know exactly where we're going with this. So that's it. And I'll take questions from the audience.
Yeah, well, the audience is me, and you answered a good number of the ones I had been jotting down. But so there, and I don't want to jump ahead to the next presentation. So, I guess if you're sort of looking at, a few times you made reference to sort of different perspective, the virus's perspective, the cell. I'm trying to put in context what you might consider an adaptation of the virus, and what is really we would call a spandrel or a side effect, And so when I'm trying to think of an analogy as you're describing these things. So, for example, it may be in some case. I think suppression of apoptosis, for example, is probably pretty clearly adaptive to the virus, right? It keeps the factory churning out viral particles, but some of the other stuff, I wonder if it's accidental side effects, like it has protein
that's a great question.
Does something, and it makes sense, but it has as far as the virus goes, it doesn't care about anything, but it certainly wouldn't care about a handful of cells in a host that it's infecting, right? So is it just kind of charging through and doing all this damage as it goes through, and there's no real benefit to the virus, but neither is there really a cost?
Right.
So how would you kind of characterize?
Yeah. So at a most basic level, for the virus to have a set of cells that are immortal, essentially immortal, right? Dividing continuously and spreading through the body, but that are also infected, is to its advantage, right? So if the virus is actually present in the cells that it's infecting, then it's only to the benefit of the virus to have cells that are immortal. So the induction of the cancer then provides the virus with a fitness advantage. The question you asked is a very interesting one. So I don't have the answer on a case-by-case basis, but I'm just speaking in general terms. So in that case, you can assume that there is a fitness advantage, including but not limited to apoptosis suppression. C-gas signaling suppression could do the same thing, for example. And induction of DNA double-strand breaks can also be adaptive. So for some viruses, they induce double-strand breaks as part of the reproductive cycle.
Thinking like a retrovirus or something. That's
correct.
Right.
So some viruses induce DNA double-strand breaks as part of the reproductive cycle, while for other viruses, what's happening is the DNA double-strand breaks are being caused because the virus is sucking up host resources to the point where replication forks are collapsing. So the cell is trying to do its own thing, dividing normally, and the virus meanwhile has sucked up all of say cytosine or adenosine, and now all of a sudden replication forks are hanging out, and replication forks, when they hang out long enough, they do cause strand breaks. So I think it's a mix. I think some of it is adaptive and some of it is spandrel. But from the virus's standpoint, it doesn't care.
No, I mean, it could charge through and leave a trail of destruction in some of the cells. It's moved on to other cells, right?
There is no fitness selective pressure to be anticipated, a negative selective pressure from having the virus not be carcinogenic in these cases. These viruses will not evolve to become non-carcinogenic.
Well, especially with the kind of time lag that you're talking about where that manifests, right? I mean, it's already been transmitted to new hosts long before, most likely. It takes years to show up.
Exactly.
Yeah. So I guess what kind of also occurs is a lot of these things seem like they would be pretty fundamental to many viruses, right? That if you think of a sort of broad categories of things viruses do to host cells, so it's going to interfere with cellular division in some form or another. It's going to suppress apoptosis, various things that are that are just obviously of benefit to the virus. Why? I guess I would put it like this: the assumption is not that all viruses are carcinogenic, or at least not detectably so in an obviously causal way. So, are there going to be patterns, do you think, in which ones are most likely to do this? I think you've hit, is it persistent is a good example. Are
Persistent is a good
retroviruses
one.
more likely, or
Yes.
yeah, and it has
So
to
it turns out
certain
retroviruses
tissues.
are a little more, yeah. Mhm. So
Yeah,
persistent in dividing tissues is a warning, is a red flag.
yeah, okay.
And then retroviruses, it seems, just empirically, they seem to be a little more prone to being carcinogenic. But
If you
yeah, again, I'm not a viral oncologist, so that the question is better aimed at them. But I think from a practical standpoint, I think, the thing about it is that we probably haven't fully tapped into the full list of viruses that are carcinogenic, just because they've been using that oncogene criterion. Um, and that's a dated criterion. So, for example, hepatitis C virus causes cancer without having oncogenes present. It's entirely possible that a number of the viruses that are on the bubble, but
Mhm.
whether or not they're carcinogenic, um, a lot of the debate is coming from the absence of oncogenes anyway. So it's entirely possible that if you kind of take a step back and look at it through through the carcinogenesis lens instead of the oncogene lens,
Right.
you end up with a different answer.
I wonder too about just the nature of the kinds of evidence. So if you're in addition to looking for genes, but if you're looking for a presence of of the virus in tumor cells or something, well
Yes,
maybe
absolutely,
it instigated this process 10 years ago and the virus has since been cleared,
absolutely.
right? So there's a
And we're going to
lag,
dig into that.
right?
Yeah,
Okay.
exactly. And we're going to dig into that in a lot of depth in a subsequent talk.
So the one, I guess I'll just,
Cool.
one other quick question about, we've been thinking, you and I have been talking quite a bit about emergent viruses and pandemic viruses that are recent experiences for the host versus ones that have been undergoing antagonistic coevolution with hosts for a longer period of time. In which case would you imagine this carcinogenic um, impact to be more significant? I get so the virus
Yes,
has
that's a
evolved
great
along with
question.
the
Yeah.
host, or it's new and the host can't suppress it. Maybe it's not an obvious prediction, but I guess what would be your best guess?
Yeah, that's a great question. I don't have a good response to that. There's a suspicion, for example, that cytomegalovirus
Hmm.
um, is carcinogenic. If that's true, that'll be kind of interesting because it's been with us, it's evolved along with us for, hundreds of millions of years, I think, if I remember correctly. So before humans existed, long before mammals existed as well. The hepatitis virus as well, I think, is really ancient. So unclear. But on the other hand, there are very recently emerged viruses where you can make the case
Hmm.
provisionally that they may be carcinogenic. I won't name names, but again, so it could be that it's just one of the side effects of viruses. Um, viruses are not there for they're not our friends.
Hmm.
So and it could be scattered randomly through it. Again, I don't see any selection pressure driving viruses to be less or more carcinogenic. It's just a feature that some viruses have and some don't.
I could see, I guess if I were going to, if I had to make some kind of guess on that, I would see that there's certainly not a selective pressure against it, unless you've got a very interesting virus that needs to be in a healthy host for a long lifespan or something, right? Like
Right.
I mean,
Yeah.
there's just probably
Sure. Sure. Yeah.
no cases like that.
Yeah, I mean,
Um,
conjecturally, one could think that maybe, one could
you could
invent a
imagine
case.
a
Yeah.
case. More likely, so on whether it could evolve to be more carcinogenic, I don't know that that would be necessarily adaptive, but I could imagine it getting in this arms race. So the longer it's either persisting in the body or longer that it's in association with that host species, you could imagine that coevolution is sort of driving more and more extreme expressions of these systems,
Hmm.
right?
Yeah.
The host ability to kind of confront that versus the virus's ability to trigger the things that are adaptive to it. And therefore, you can imagine the side effects might get worse over time. So, for example, you could have a virus emerge that has potential to do this,
Hmm.
and
Yeah. Yeah.
in fact, over a number of years, it might actually get worse at doing this.
Yeah.
So there's certainly no case where you would assume it's going to get better, right? It's going to become more, but
Right.
I don't think
Yeah, no, that's a
but
great point.
I can see a case where it could get worse as there's more of
Could
it,
get worse. Yeah, totally agree. All
especially
right,
if it
great.
evolves to be more persistent.
No, I agree. Yes, that's a good observation. All right,
Very
great. Well,
interesting.
so this was a good time, and we'll be back
Yeah,
shortly
we'll be
for
back, and I think
part
we're both
three.
kind of chomping at the bit a little bit to follow through what I'm sure a lot of the viewers have already surmised, is putting part one and part two together in the context of current events. So yeah, we'll look forward to that, and we'll hopefully get that posted soon as well.
Sounds good.
All right.
Thank you.
Awesome. Cheers.
Bye.